recording in the middle of a session, it doesn't record whatever you did before then. So I'm recording now. Sorry that I didn't record before. Cardiac output, stroke volume, heart rate, SV, AHR, CO. Okay, there we go. We're back. So beta receptor activation is going to increase heart rate by increasing the permeability of sinoatrial node cells to sodium via the funny current channels, which are cyclic nucleotide gated, and beta receptors increase the concentration of CAMP in the cell. Now that's a whole bunch of physiology there in 2 seconds, but you got all that because y'all are smart. Stroke volume. Stroke volume is just how intensely the heart pumps. And as we'll see, beta receptors on the heart, well, we've already talked about this. Beta receptors on the heart not only increase the speed of depolarization of the sinoatrial node cells, they also increase the contractility of the contractile cells, or contractive cells. So not only are they going faster, they're pumping even more, like they're kind of squeezing really hard when beta receptors. So beta receptors are going to vastly increase cardiac output by increasing both heart rate and stroke volume. Now, when we talked about smooth muscles, we talked about smooth muscles around veins, because you've got the veins, you've got your intima, and then your muscularis, and then you have some adventitia around it. It's not very thick because veins need to be able to really stretch out, because remember, veins they hold on to most of the blood in the body, about 60% of the blood. But this smooth muscle here, the muscularis layer, and these are called tunics, so tunica muscularis, squeezes down with alpha-1 receptor activation. Alpha-1 receptors, remember, are adrenergic receptors. They respond to norepinephrine or noradrenaline. Norepinephrine and noradrenaline are the same thing. If I said that and you thought, oh my God, is norepinephrine or noradrenaline? Same exact thing. So alpha-1 activation, these squeeze down. So when you squeeze down the veins, you decrease the size of the veins. What that's going to do is it's going to squeeze a lot of the blood that's held in the veins back into the right atrium. It's going to go to the right ventricle, to the lungs, to the left atrium, to the left ventricle, to the aorta. Functionally, what you're doing is you're increasing the volume pretty drastically of the blood in the systemic circulation. Well, not in the systemic circulation, in the arterial circulation. So you're increasing the amount of blood that's now entering the aorta. So if we go back and look at the Wiggers diagram, we have aortic pressure right here. That's going to go up even higher if you have activation of the sympathetic nervous system. And that's going to happen for multiple reasons. One, sympathetic activation increases heart rate, increases stroke volume, increases venous return. So you're increasing venous return, you're increasing stroke volume, you're increasing heart rate. All of this is going to increase the amount of blood that flows into the aorta, which is all going to increase systolic blood pressure. And systolic blood pressure, remember, systole is contraction. You have an atrial systole and a ventricular systole, but when we refer to systole without a prefix, we're talking about ventricular systole, which is represented by the QRS wave and then comes in this purple space here, and there should be ventricular systole. Yeah. So this volume is going to go up. So it's going to go up to here or so. When you have an increase, I drew a line there, but it didn't come up. When you have an increase in, there it is, right here. When you have an increase in sympathetic drive or sympathetic activation. Now, there are also some alpha-1 receptors on the arteries. Not so much on the, there are very few on the aorta, because the last thing you want to do is really squeeze down the aorta and hold blood in the ventricle. So the aorta is large, but say in your carotids, you can squeeze those down. In your subclavians, really what you see is in different parts of your body, especially in the celiac artery, in the mesenteric arteries, they're really going to squeeze down with sympathetic activation, because not only do you want to increase pressure there, you want to decrease flow to your viscera when the sympathetic nervous system is activated. So when you increase all of these, you increase something called mean arterial pressure. Mean arterial pressure is calculated by systolic pressure plus diastolic pressure. I'm going to have to start abbreviating. I'm going to have to move things. Oops. Diastolic pressure times 2. And the reason it's times 2 is because the diastolic phase comprises 2/3 of the heart cycle. And if we look back at our Wigger's diagram, from here to here is about 450 milliseconds. And from here to here is about half that, 225 milliseconds. So that's why you double the diastolic pressure. And then all of this divided by 3, because you have 1, 2, 3. And that's going to give you the mean pressure in the artery. Meaning, okay, for 250 milliseconds, the pressure in the arteries is 120. For twice that long, it's about 80. So mean arterial pressure for our stereotypical 120 over 80, that would be 120 plus 160, which is 280. Is that right? Yes, 280 divided by 3 equals, what is that, like 93? I think that's right. 280, because 270 divided by 3 would be 90. So that'd be about 93, 92.333. Okay, so you'll see mean arterial pressure, abbreviated MAP, and this is a measure that clinicians will take. A lot of times you just write systolic over diastolic, but mean arterial pressure can just give you a one number idea of how much pressure there is in the arteries. Okay. Now, we talked about when we in my lecture on smooth muscle, and you should really go back and look at that, because alpha 1 receptors, like I said, alpha 1 receptors are GQ proteins, and what they do is they cause smooth muscle to contract. Beta receptors, they do the opposite. These are GS receptors, and they cause smooth muscle to relax. And this is through the intracellular cascade that pits MLCK against MLCP. That's a kinase and a phosphorylase, which adds or takes away a phosphate group to myosin light chain, which is the regulatory protein on the myosin that's found in all types of muscle. But it's just a regulatory subunit. And in muscle, it actually innovation involved in regulatory protein. It's not phosphorylated to a great degree. Even if it is, myosin cannot bind to tropomyosin, the myosin binding site of actin. So what we're doing here basically is taking all these microcells and applying to large processes with Apple Watch, what is the thing when you walk, or a sphygmometer, which is for blood pressure, pulsometer is how you measure heart rate, and measuring with a pulsometer your pulse, what your is arterial pressure, but truly is that you feel when you years over about 4 millimeters of mercury. That's about the pressure you're feeling, because all you're feeling is the difference between the diastolic and the systolic pressure. So it's that difference that you feel. You're starting right here, kind of moving up and down from 120 back to 80, 120 back, 20 back to 80. So the flow of blood is proportional to, this brings back cardiac output, is proportional to your cardiac output minus your arterial resistance. So the blood flow into any part of your body is going to depend on cardiac output, which depends on the strength of contractility and rate of contractility of your heart, and then the tightness of your arteries. however resistant they are. The veins, they don't have any pressure at all. There's no pressure from the heart in your veins. Sorry about the beep, and I meant to close my mail, but haven't yet. So the mean arterial pressure, the flow is proportional to cardiac output minus arterial resistance, and mean arterial pressure is proportional to cardiac output times the resistance. These are in opposition. One is directly proportional to arterial resistance, that's pressure, and one is inversely proportional to arterial resistance, and that's flow. And that was summed up in a little equation that I drew, oh wow, it seems like 3 years ago. Here. It's not really an equation, but flow, velocity, and pressure. The first time I wrote that, flow down with resistance and pressure up with resistance. I think if you think it kind of makes sense. The amount of pressure system, that's resistance to flow. Sympathetic nervous system increases pressure and decreases flow almost always. There's one time that it doesn't. It can actually decrease pressure and increase flow. That is receptors. You have receptors. Is everybody in the middle of the road beach? Hello?
You're cutting in and out, but it might just be my stuff.
Yeah, you're cutting out of your computer too.
Okay.
I am cutting out. Shoot. Let's see. I'm gonna take my video away. Maybe that'll help me catch up. I wonder if I hang it, that'll stop or it'll all be in me if I hang up one. I'm gonna hang up one computer. If I leave all day long, sorry. Can everybody hear me?
Yeah.
Yay! Okay, good.
Yes.
I just left the meeting on my computer. Can you see my screen now where it says beta receptors in arteries?
Not yet. Oh, now. Yeah.
Is it there now? Yes? No? Yay. Okay, see, I'm running 2 computers, and I think that really clogs up my internet. Part of me didn't want to come home for that reason and just stay on campus because that internet's a lot quicker than mine. Maybe I should call Comcast and just suck it up and get faster internet for the next month. I should probably do that anyway. Okay, so beta receptors in your arteries. What do beta receptors do to smooth muscle? Does anybody remember that from 90 seconds ago?
They relax.
Yes, exactly. So they relax smooth muscle. So that's going to decrease resistance and increase flow. So where do you think you would find beta receptors in arteries? When the sympathetic nervous system is activated, where is it that you need to increase the flow of blood?
To the brain and heart.
To the brain Yep. The heart. What do you need to do when a baboon jumps out of a tree and chases you? You need to be aware of it. Your heart needs to increase.
Muscles.
Yeah, your muscles, like your leg muscles. And your sympathetic nervous system is it's not the smartest thing in the world. You just activate everything. Your digestive system shuts down. You have alpha receptors on the blood vessels, on the arteries leading to your digestive system, but to your bicep, to your tricep, to your gastrocnemius, to your semimembranous, semitendinous, biceps femoris, all of your quads, all those muscles increase blood flow. So those beta receptors increase to your. What that's going to do is going to deliver more glucose. It's going to deliver more. It's going to deliver more oxygen. Muscles. And a really interesting thing is that in addition to the sympathetic nervous system and norepinephrine, on these receptors, I always need to remember that I do spend about 3 hours a day talking, because at like 1 o'clock, I've always think I've got corona because my throat hurts. Know my neuroses. Oxygen. So oxygen receptors on the smooth muscle in arteries, that's going to cause blood vessels to dilate. There's also carbon dioxide dilate vessels. And you can see why this is a adaptive response, because when your muscle is hypoxic, the first thing you want to do is deliver more blood. Now, there are a number of ligands that also, and I just alluded to this when we were talking about smooth muscle, but this is really active in the cardiovascular system. Serotonin, serum, tone. That's what serotonin means. That's why we don't call it 5-hydroxytryptamine. We call it serotonin, which is 5-HT, because it tones up the things that carry blood serum. Arginine vasopressin, or AVP. Norepinephrine and epinephrine, we already talked about those. These are all vasoconstrictors. So they are going to act through a membrane-bound receptor, typically a G protein, to cause blood vessels to constrict. Another way you can constrict blood vessels, so we talked about oxygen, carbon dioxide, serotonin, vasopressin, sympathetic nervous system. I mentioned in the smooth muscle discussion that you have a myogenic reflex. Go back and listen to that. The myogenic reflex, it's called this because the flexion is generated by the muscle itself. So it's intrinsic to the muscle. When the smooth muscle goes from this big around to this big around, it stretches the sarcolemma, the cell membrane, and that stretching causes calcium channels to open. Calcium pours in, binds to calmodulin, which activates or upregulates the activity of MLCK, which then phosphorylates the myosin light chain on the myosin, causing the flexion of the elements in the smooth muscle. If you eat a lot of salt, that's going to increase sodium concentration in the body. Increased sodium concentration leads to the production of renin by the kidneys. And we may talk about the kidneys this semester. I don't know if we'll get there. It's one of my, so maybe we will get there. But renin causes a cascade that leads to the production of aldosterone, a mineralocorticoid. And this causes water retention. You increase the amount of water in your body, you're going to increase the amount of blood in your body because that's where most of the water is held. And this is going to increase blood pressure because you're increasing the actual amount of blood. I should say, does anybody know what the between renin and aldosterone? You've probably heard. I know one of you has paper on it. Angiotensin. Tense means pressure, tense up. Angio means blood vessel. Angiotensin, in addition to activating or causing the release of aldosterone, which is going to increase water retention, which increases blood pressure, angiotensin acts directly on smooth muscle, causing it to constrict. So it's all two different ways angiotensin to lead to an increase in the angios or blood vessels, and it causes sodium retention, which then causes fluid retention, which regulates blood pressure. Okay, I'm going to one more thing, which is one of the cooler things about vascular physiology, I think. And it has to do with the capillary beds. So you've got your nice, bright arteries here. And they flow into capillaries. You've got your capillary bed here that then flow back into vein. Okay, so the arteries flow into capillaries, which flow into veins. And at the arterial end of the capillary, you have some residual. So you have some residual blood pressure. Because here your blood pressure is 120/80. It diminishes a bit because now we're down at the capillaries. So the hydrostatic pressure is around 32 millimeters of mercury once you get all the way down into the capillaries, so down here. And it's diminished from 120/80 because what you're dividing and dividing and dividing and dividing, and these are little tiny arterioles going into capillaries. Now, what happens at the capillary beds is you have a lot of leakage, fluid comes out, bathes all of the tissue in glucose and hormones and all of the things that are traveling in the blood, cytokines, what have you. What that does is it decreases hydrostatic pressure until you get over here down to about 15 millimeters of mercury. Does that make sense? It's kind of like going through a soaker hose. You lose a lot of the pressure. I'm going to take the silence as, yes, that is crystal. So if we were to draw a graph, and we have hydrostatic pressure, it really should be hydrodynamic pressure, but because this is a moving system and a dynamic system. I don't know why isn't it? Oh, there we go. Okay, so hydrostatic pressure is going from 32 down to 15. I don't know why I'm writing in big fat marker. There's another pressure, which is osmotic pressure. And we talked about this at the beginning of the semester. Osmotic pressure is mediated by large, we call them colloidal proteins. If you remember, these are albumins, fibrinogens, and what is the third colloidal protein? Somebody help me out here. I don't know why I can't remember this word. You're going to make me Google this? Oh, I'm not hooked up to the internet on this computer. Globulins. Immunoglobulins, remember. I can't believe y'all made me look that up. So,
Sorry.
that's okay. Thank you for the apology and the recognition that somebody's listening. So if you remember, the albumins, fibrinogens, and globulins do not leave the capillaries. They're too big. That's one of the things that's different. If you go back and look at that one of the first graphs that we looked at in this class, and it was the difference between sodium, potassium, magnesium, and chloride in the intracellular fluid, extracellular fluid, and the extracellular fluid is divided between blood plasma and interstitial fluid. The one difference between blood plasma and interstitial fluid is that there are tons of proteins in blood plasma. Those are these guys right here. And there's really not many proteins at all in the interstitial fluid because they sit at an osmotic pressure of about 25 millimeters of mercury. And it's constant as you go from the arterial end of the capillaries to the venous end of the capillaries. Does this graph make sense to everybody?
Yes.
Okay, wonderful. Thank you. Arteries, and then this is veins. But this is arterial end of the capillary and venous end of the capillary. Now, what you see is the hydrostatic pressure surpasses the osmotic pressure. And this facilitates, or you could say it enables, it makes stuff go out. It facilitates outflow. So what happens is all of this fluid goes out because the pressure in the capillary bed is higher than the pressure outside of the capillary bed. Here, the hydrostatic pressure is 15 millimeters of mercury. The osmotic pressure is higher than that. So this here at the venous end, it facilitates inflow or absorption. So at the arterial end, what happens is things come out of the bloodstream. Things filtrate. They go out of the bloodstream into the interstitial fluid. Once you get over here, as you move from the arterial end to the venous end, those capillaries are going to start sucking things back in. And what it sucks in is waste material, sucks in carbon dioxide, lactic acid, metabolites of hormones, anything that the interstitial fluid needs to get rid of is going to be in high concentration and flow into the cell. That doesn't happen at the arterial end because the hydrostatic pressure has surpassed the osmotic pressure. Now, if you're malnutritioned, if you don't have enough food, what happens is this line right here falls down. We'll draw it a little bit darker to show that you're malnutritioned. It can get suppressed down to say we'll just say 12 millimeters of mercury. And this is because you're not eating enough protein. You're not taking in enough protein. And what that does is it decreases osmotic pressure below the hydrostatic pressure, and that doesn't facilitate inflow into the capillaries. All of that fluid stays out of your veins, goes into the interstitial fluid, and you end up with a big, huge belly. And this is called abdominal edema. And you see this in individuals that are highly malnutritioned. When I was in high school, it was Ethiopia. Now it's Yemen, which is, I read an article this morning. That's probably one of the, not one of the few, but one of the things that's beneficial about the whole corona outbreak. It seems to be stopping I think Saudi Arabia called a ceasefire on their war in Yemen. That's kind of a good thing. Okay, I think that's really everything I want to talk about today, and it's almost time to go. Anyway, we've got 10 minutes left. I am happy to talk about anything that you may have missed in class, or just that you want to mention, ask, talk about. Anybody have any questions, concerns, ideas? Y'all are all bright and wonderful people, so
The quiz that you're going to post today that's due on Tuesday, does that quiz count as one quiz for next week or a quiz for this week?
that'll be a quiz for
If that makes sense.
this week. Yeah.
Okay.
Yeah. So I'm basically what I'm trying to do is get points in for y'all. That's my goal. It's not so much that I want to have 3 quizzes a week. It's that by the end of the semester, I want to have 1,200 points so that things really count what you thought they would count. So, I mean, it's not a decision that I think y'all make all the time, but if you made a pragmatic decision that, okay, I'm gonna bomb this lab report because it's only 50 points and there's 1,200 points in the class, so that's only whatever % of my grade. I can do that. That's why I try to stick with the 1,200 point idea, because then if I reduce the points to 800 points in the class, then your calculation earlier, I don't know, that's a very pragmatic calculation. I'm not sure any of you think like that, but if you do,